Today's mini lecture is about a process that DNA uses called transcription. Transcription will convert a gene found on DNA into a single-stranded molecule called RNA. RNA is very important because it carries DNA's instructions. There's a thing that we're going to learn about called the central dogma, and it states that information flows in one direction only, from DNA to RNA to proteins. Now, the central dogma includes three processes. Replication, which we've already learned about, is the first process, and this is where DNA can make a copy of itself. The next process that we're focusing on today is called transcription, and this is where DNA is used to make a molecule called RNA. Then finally, what we'll be learning about next class is a process called translation, and this is where an RNA molecule is used to finally make proteins. So RNA is going to be the link between DNA and proteins. Now I want us to hone in on some keywords here. The first one is transcription. This is made from the word transcribe. Transcribe is a very fancy word that just means to rewrite, and this is very important because DNA, the language that it uses are nucleotides, okay? The language that RNA uses are also nucleotides. So basically, transcription kind of rewrites something in DNA. Translation, however, comes from the word to translate. Think about maybe going from, say, English to Chinese. You're completely changing the format. You're changing the language. Similarly, we go from nucleotides, which we've used in RNA and DNA, all of a sudden to something called amino acids. So you're completely changing the language, so to speak, that the information's being carried in. And we'll learn more about these processes in depth. So RNA, or ribonucleic acid, differs from DNA, or deoxyribonucleic acid, in three major ways. First, RNA has a ribose sugar, whereas DNA has a deoxyribose sugar. Second, RNA has the nucleotide uracil instead of thymine. And finally, RNA is a single-stranded structure, whereas DNA is a double-stranded structure. So let's take a look at what this process might actually look like. So transcription copies DNA to make a strand of RNA. The main thing that catalyzes or starts transcription is called RNA polymerase. It's very similar to DNA polymerase. It's an enzyme. So the first thing that happens is RNA polymerase and these other proteins and enzymes form something called a transcription complex, which is that big yellow bubble you see below. The transcription complex recognizes the start of a gene on the DNA and unwinds a segment of it. Next, free-floating nucleotides will pair with one strand of the DNA. RNA polymerase bonds the nucleotides together, and the DNA helix winds up again as the gene is transcribed. Finally, when it's done, the RNA strand will detach from the DNA. Transcription is used to make three types of RNA. The first is messenger RNA, or mRNA, and this is probably the most important because this is what carries the message that will eventually be used in translation to form a protein, and that is shown down here. Next, we have our ribosomal RNA, or rRNA, and this forms parts of ribosomes where the proteins are made. And then finally on this side, we have our transfer RNA, or tRNA, and this is important because it brings amino acids from the cytoplasm to a ribosome. The transcription process, you may have noticed, is very similar to replication. Transcription and replication both involve very complex enzymes and use complementary base pairing rules. The two processes, however, have very different end results. Replication copies all of the DNA. However, transcription will only copy a gene, or basically what it needs to make a protein. Replication also makes a single copy, whereas transcription can make many copies. That concludes this lecture. If you have any questions, please let me know.